Hi guys, Matthew here. Today in this lecture we're going to talk about temporal analysis. So the textbook has on the study of plant disease epidemics has 3 chapters on temporal analysis, which the first one's chapter 4, 5, and 6 are on temporal analysis. Then it'll have 3 chapters on purely spatial analysis. And then it has some chapters beyond that mix a bunch of these ideas. So today we're just going to talk about quantifying and comparing epidemics. Then the next chapter, what's the next chapter on? I don't remember, some other topic in time analysis, and then there's another chapter. Anyways, so chapter 4, temporal analysis 1. Analysis 1. The subtitle is Quantifying and Comparing Epidemics. So the main goal of this chapter is just to introduce you to the models that they use to quantify epidemics and some of the parameters that they commonly use. And then it talks about comparing epidemics. I'm not going to go real in depth into that. I know that's important, but I'm not going to lecture on that. So why do we care? Because epidemics, by definition, are a change in disease intensity over time and space. And in this section, we're just looking at time. Well, a lot of useful information can be gathered from an epidemic by just looking at it as a function of time. And it's much easier to handle. When you do space, you have 1-dimensional space, 2-dimensional space. You can get into 3-dimensional or complicated shapes. And then if you mix in time with that, how the disease travels and spreads over time and space and gets really complicated. So we're starting simple. Time's 1-dimensional, real simple, easy to handle, and it can actually give us some really useful information about the disease progress curve, or about to quantify how the epidemic changes over time. So the main graph that's used a lot in this chapter is they call the disease progress curve. Disease progress curve. And we have time, T, and we have intensity. Intensity. Typically denoted by a lowercase y. And probably the most common shape is the sigmoid shape, but it can take on a variety of shapes, so they're not all necessarily of that shape. The intensity is whatever measure you're looking at. A common one in this chapter is the proportion of disease plants. So if it's a proportion, it ranges from 0 to 1. If it's not a proportion or a probability, they use the capital letters to denote them, and that would be like the count, the total number of disease plants, or the average area per meter squared of disease plant, or something that's not really a proportion but some other value. And I'm not going to talk about that. If you want to learn about it, you can. Basically the exact same stuff applies to this or that. All you do is add a scaling factor to the graph, and that's pretty much it. The goals. So we can do this to determine if 2 epidemics differ. Common one is you give something a treatment, and so this is the curve for the control, and then this would be the curve for the treatment. Say you want to know if your fungicide works. You invented a fungicide and you spray it on one field and then you don't spray it on the other field, and you measure how the disease intensity increases over time, and then you want to know if your fungicide's working or not. So you can compare these 2 graphs. That's one use of this stuff. Another use is just to quantify stuff. You just want to know just some basic stats. You just want to see what happens. You don't really have a goal. Another goal, though, is to quantify it so in the future you can predict what will happen. So then you get a disease progress curve and you say, well, this is what happened in my field. So if someone else gets this disease, we expect it to behave similar to what happened in my field. So it can be used for prediction. can also be, you could derive or come up with some theoretical idea of how the disease spreads and progresses and whatever, and you want to see if your theory matches the real data. So you collect data and then you plot intensity over time, and you can use this to validate your theoretical model. You can also use it to evaluate and identify factors affecting the magnitudes and rates of the disease progress curve. Blah, blah, blah. One of the main goals is to just select the appropriate model to characterize the epidemic with a small number of parameters. We want to keep the model as simple as possible, so we want as few parameters as we can. We want to simplify it as much as it can be simplified for our goal, but without simplifying it more than that. So then some comments. None of these models ever fit real data perfectly. There's always something called unexplained variability, also known as error, and that's just part of life. And these are done with computers to fit the model. Most of the stuff you can understand it in theory. You can derive these equations by hand, relatively easy, but to actually do this in practice, you need a computer. Because when you get a bunch of data points, more than 30 to 100 data points, it just gets unmanageable to do by hand. And so if you are just interested in comparing two curves, you want to know if the curves are significantly different or not, you don't even have to go into this complicated modeling stuff we're about to do. There's two techniques. One's called repeated measures ANOVA. It'll tell you if this curve is significantly different than this curve or not. The other technique is known as area under the disease progress curve, and you can do a hypothesis test to see if they're different or not. And I'm not going to lecture on those, but if you want to just compare two curves, you can Google repeated measures ANOVA or AUDPC, area under disease progress curve. Then I don't know if they use this or not, but when I took survival analysis, there's a bunch of methods developed to compare curves, and they would calculate confidence intervals about the curves, and then see if the confidence intervals overlap. And they could do that for proportions too. You might be able to do that, but the whole idea in curves comparing, you sort of from my survival analysis background, which this is different than the repeated measures ANOVA, but you basically have a confidence bound. You know the true curve somewhere in here, and so we get this area where it's likely the true curve, because we just approximate the curves with data, but the true curve's in that zone, and this one, the true curve's in this zone. And then we note if these two confidence bands around each curve, if the bands were overlapping, then there'd be no evidence that the curves are different. But in this picture, the bands aren't overlapping for much of these curves. So we say because there's a huge area where they're not overlapping, the curves are significantly different. But if the bands did overlap for most or all of it, then we'd say that there's not strong evidence that they're different. But then repeated measures ANOVA, it sort of does that kind of idea, but it does it point-wise, right? And it averages out the total difference between the height between points and averages that across all points, I think, and then gives you a result. And the area under the disease progress curve compares the two areas and normalizes it and gives you a result. And none of those require assumptions of models. It just is comparing shapes. Okay, so let's get into some general concepts. Epidemics are dynamic processes, and dynamic processes are typically represented by rates of change. So most of what we'll be doing today is concerned with the dy dt thing, the change in intensity with respect to time. So the rate of change of intensity with respect to time is represented by dy dt, can also be written as dy of t. dt. And a lot of the stuff is, we rarely are interested in this. We're not so much interested in y of t. We're more interested in the change in y of t. Because epidemics are defined as the change in disease intensity over time and space. So we're interested in the change. We don't care about any particular time. We just care if it's increasing, constant, decreasing. That's blah blah blah. There's a bunch of other stuff. Disease progress curves. Let's go back to that, revisit that real quick. A disease progress curve, abbreviated DPC, disease progress curve. They are typically increasing monotonically, like exponential, monomolecular, sigmoid, some other shape. But if you notice, they're always increasing. That's not always the case. There's a handful of examples where it can actually decrease. If the plants are reproducing faster than they're becoming infected with diseases, then you can get something like this. I don't know how it works, but the Dutch elm tree is known to, there's some disease that affects the Dutch elm tree, and a huge proportion of them can get disease, and then they'll reproduce, and now they had 1,000 trees here, they reproduced maybe 500, so now the proportion of diseased trees drops, even though the total number of diseased trees has not decreased, but the proportion of diseased trees has dropped. That's something interesting. Anyways, disease progress curves in theory can assume any shape you can make with a curve, but realistically, they sort of tend to be some type of monotonically increasing shape. And then also there's a relationship. The intensity y is the integral of what we're interested in, the rate of change dy dt with respect to t. So how do epidemics occur? The inoculum, we're going to call it an infectious unit. Each little element that can infect a tree is called an infectious unit. So an example would be a fungus produces a bunch of spores and they go float around in the air till they hit something they can infect. Each spore would count as one infectious unit. Some types of inoculum are fungus, spores, also the fungus. Sometimes a fungus can infect something. It doesn't have to even be sporating or whatever that word is. And then bacteria and viruses. I think that's all. And then viruses, and in the case of the Asian citrus psyllid, bacteria can be transmitted by vector, or a vector is the method which it's physically transmitted. But most of the viruses or all are transmitted by insects, and the bacteria can be transmitted by insects or blowing in the wind or in the water or whatever. There's different ways they can physically move from one location to another. This is called the vector. Then there's two major types of epidemic classifications as far as this chapter is concerned. And there's what are called polycyclic and monocyclic. So polycyclic and monocyclic. In a polycyclic epidemic, they typically spread from plant to plant. As the epidemic's occurring, a lesion will form, it will produce spores, infect a new area, create a new lesion, produce more spores, blah blah blah. And that's polycyclic epidemic. A monocyclic epidemic, this isn't going to be a real good definition, but basically the inoculum is not released or available until some later time, such as after the plant starts to die and decay, or at the end of the growing season, or it must be transmitted by bugs from one species of plant to another, and it can't be transmitted within species, or something like that. And the reason why this distinction is important is the models used are different. Polycyclics use a lot of the exponential type growth models. The monocyclics use a lot of the monomolecular type growth models, and you tend to use different growth models. And because they have different growth models, there's different ways you control for disease depending upon what type of epidemic it is. And so some examples of polycyclic are the potato lapelite. It's constantly producing new spores and reinfecting it, and it's constantly ongoing. And it increases very rapidly, and that's what happens. But then in monomolecular, something like maize smut, which I don't know much about, but it can only spread at the end of the growing season. So during the growing season, it doesn't spread at all, but when you're at the end of the season. So when you're harvesting it or whatever, at that point in time is the only time it can spread. And that causes it to behave differently. Another example is verticillium wilt of potato. The potato is infected and it's producing inoculum probably continuously, I don't know. But the inoculum doesn't affect the surrounding till the plant starts to decay, because it's all contained inside the plant, and it doesn't get to float around and infect other stuff till long after the plant's dead and already decaying. And that causes the disease progress curve to have a different shape. These typically evolve very slowly over time. These evolve very rapidly, and these you treat differently. They can be controlled by different methods such as crop cycling and whatever, and sometimes those methods don't really apply to them because of how they work. So we're gonna go over 5 different models I meant. So the models we're gonna cover, and I'm not gonna go in really much detail, I'm just gonna look through them. If you want to learn more about them, Google them. The exponential model. All of these have the word model after it, but I'm not going to keep writing the word model, because, whatever. I'm lazy. Monomolecular logistic. These names might be kind of scary, but these models aren't actually all that scary, so don't be scared. Gompertz and Richards. Voila. Okay, let's go into them. So the exponential model, you all have probably heard of or seen an exponential growth model, and so shouldn't be too new to you. Exponential model. Exponential models aren't used in just epidemiology, they're used in finance. It's called exponential compound interest when you do interest calculations for loans, which I think compound interest is kind of bullshit because it can really screw you over financially. But anyways, compound interest is an exponential growth model. It's the growth of populations of like human beings or whatever. It's the exponential growth model. A bunch of other examples of it too. So blah blah blah. So we define it in terms of the rates because that's really what we're interested in. So the rate of change in the disease intensity with respect to time is equal to a rate constant, R sub E. The subscripts stand for the model is referring to, because all of these models have a rate constant. So they're all going to be R sub something times Y. So it's the rate constant times Y. So the proportion of new infections is equal to constant times the number of current infections. So that's what it says. New infections is equal to constant times current infection. Then if we solve for y, because we feel like doing a bunch of math, but first I'll draw the graph. So this is time, this is the proportion of infected plants, intensity y. They have that shape. Depending on what this R value is, they have different steepnesses or whatever, but they still all have that general shape of curving upward. So let's derive what y is equal to, because I feel like doing some math, and I was proud that I figured this out a little while ago. Hopefully it fits on the board. So this is this. We can do what's called separation. This is a differential equation. This is a derivative. You can put it in differential form. I'm not going to. You could do dy is equal to this times that, dt would be a differential, whatever. Do separation of variables. Then we get dy over y is equal to r sub e dt. So we have all the y's grouped together and all the non-y's grouped together. You could put the r sub e anywhere, it doesn't matter, it's constant. Whatever. Then we integrate and integrate. Whee! I used to think differential equations when I took the class, it was like mind-blowing. And now I'm doing it right now and it's like super not mind-blowing at all. Then I'm just gonna do indefinite integrals, no limits, because I don't feel like going into definite stuff. This is equal to natural log of the absolute value of y plus a constant is equal to r sub e times t plus a constant. I'm going to group this constant in that one. Then y ranges between 0 and 1. If we restrict y to always be greater than 0, this absolute value symbol can go away and can just be parentheses, because this only matters if y is negative or 0, and it's not when making that restriction. It's actually there. Then we don't need absolute value symbols because it's never going to go negative. Is equal to r sub e times t minus c. Move the c over there. Then do e to both sides. We get y is equal to e to the r sub e times t times e to the negative c. Is that really negative? Guess so. And we're just going to call that k. So y sub 0 is equal to k e to the r sub e t. Then I'm going to do some erasing just to finish it up. We're going to go up here. We're at y is equal to k e to the r sub e t. And we know that y at t equals 0 is called y naught, the initial proportion of diseased plants. And that's equal to k e to the r sub e times 0. E to the, or it's this times that's 0, e to the 0 is defined as 1. So 1 times k is just k. That means k is equal to y sub 0. So then we get y is equal to y sub 0 e to the r sub e times t. Voila. In case you were interested. Nothing too special. Okay, so the next model is called the monomolecular model. And here's monomolecular. Whoa. The name supposedly derives from someone who's using it for molecular chemistry stuff. But the equation is d of y dy dt is equal to r sub m 1 minus y. So if we remember in the exponential, dy dt is equal to a rate times y. In this case, it's a rate times 1 minus y. And 1 minus y is the proportion of non-infected, the proportion of healthy plants. The y is the number of diseased plants, 1 minus y is the number of healthy plants. So this is saying the rate of increase is equal to a constant times the proportion of healthy plants, which is opposite of exponential. So we have time, y, and this makes that shape, which is the opposite of what the exponential was, because exponential, this is the exponential shape, and this is monomolecular, sort of just like a mirror image about the 45-degree line, and it gives a different shape, and which is important when you note this is used a lot for monocyclic diseases, because at the end of the season, like with the maize smut at the end of the season, all of them have a massive amount of inoculum they built up, and it all releases at once, and then just immediately infects everything at once, kind of thing, versus it slowly producing inoculum and slowly infecting more with like a fungus sporation thing. And so anyways, that's monomolecular. You can write in the form r sub m. If you distribute it, dy dt is equal to r sub m minus r sub m times y. I don't know why I pointed that out in the book, but it did. If we want to solve for y, this is a differential equation. Then I go back to the original form because I like it better. We can solve for y by separate variables and integrate. So separate and integrate. So then we get dy divided by this y term. Dy divided by 1 minus y is equal to r sub m dt. So we separated variables. So it's all the y stuff, all the non-y stuff. And then we're going to integrate it. R sub m is a constant, so I'm going to leave it out there. And then we're going to integrate it symbolically, or indefinite integrals. Then integral of 1 over 1 minus negative y is equal to 1 divided by the constant in front of the variable, which is negative 1, 1 over negative 1, natural log of the absolute value of 1 minus y, which is just that term, plus a constant of integration is equal to, because we're doing it with indefinitely, is equal to r sub m times t, plus a constant, but I'm grouping the constant over there. Then we're going to make note that this is never going to be less than 0, less than or equal to 0, because we're doing the restriction 0 is less than y, which is less than or equal to 1, which makes sense because that y naught has to be at least, y naught is at least 1 over the sample size. You have to have at least one infectious individual at the beginning. And so y naught can never be 0. And if y naught can never be 0 and it has to increase monotonically, this sort of makes sense. And so clean this up. Negative natural log of 1 minus y, getting rid of the absolute value symbol, plus c is equal to r sub m times t. I'm going to erase a bunch of this stuff. And then we have y is equal to negative natural log of 1 minus y. Oh wait, what did I do? Screwed that up. Negative natural log of 1 minus y plus c is equal to r sub m times t. So move the c to the other side, multiply everything by -1. After a while. Then exponentiate everything. Then you get 1 minus y is equal to e to the c times e to the minus r sub m t. I did e to the c plus becomes multiplication times that. We're going to call this k. So k e to the minus r sub m t. Then solve for y. You get y is equal to 1 minus k e to the minus t. So now you have y is equal to 1 minus k e to the minus r sub m t. And you want to know what k is equal to. And when I first looked at this, I got down to this far when I was solving this problem a few minutes ago. I was like, couldn't figure out how to come up with k. And I saw what the answer was, because it's just, when I was reading the book, it's right there. But I'm like bashing my head against the table trying to figure out how I get that answer. And then once I realized how I get it, it was like blatantly, it was so simple. I felt retarded. So y at t equals 0 is equal to, defined as y naught, the initial number of diseased individuals. And that's equal to 1 minus k e to the minus r sub m times 0, because t is 0. This whole term, 0 times that's 0, e to the 0 is just 1. 1 times anything is just that, so k times 1 is 1, k. That's equal to 1 minus k. So then we have y sub 0 is equal to 1 minus k. Solve for k, and that gives you k is equal to 1 minus y naught. So then we go back here and plug that into the original equation. Replace k with 1 minus y sub 0 e to minus r sub m times k times t. I'm mixing up variables. Voila! That's monomolecular. So the next one is logistic. I'll spare you the derivations because these just get messier as we go, and I would probably screw them up if I tried to do them. Logistic model. Actually, I derived the logistic one before I remember doing it in college. I don't think it was that bad. dy dt is equal to r sub l times y times 1 minus y. So t y 1 0. So logistic functions have the classic sigmoid shape, sort of like an S curve, and they use the rate of change in logistic functions a proportion of the currently diseased infectious ones and the non-diseased healthy ones, and that gives it that cool shape. This comes up a lot in the biological settings, this type of shape, because when there's a low proportion of diseased plants, you can produce spores or inoculum units, spread them everywhere. It's very likely it's going to hit something that can make disease, so the disease spreads very quickly and has this exponential increase at the beginning. But then once it gets harder and harder to find non-diseased individuals, because you're just spreading inoculum and it just keeps landing on diseased individuals, it's not infecting anyone new, then it sort of starts to taper off, because it's getting harder and harder to actually find someone who's not diseased. And so then you get this is like the monomolecular part. So you have like the exponential curve at the beginning and the monomolecular slow down kind of at the end. And you do a bunch of algebra, differential equations or whatever, you eventually come up with y is equal to 1 divided by 1 plus 1 minus y sub 0 over y sub 0 exp minus rate of the logistic times time. Voila! It's kind of ugly, but it's not that bad. Then if you graph the absolute value of the rate, dy dt, the absolute value of the rate of change versus time, you get this symmetric bell. That's supposed to be symmetric. You get like a symmetric bell-shaped curve. It's not actually a bell curve, I don't believe, but it looks like it, similar to it. Pretend this is symmetric. You get that kind of shape. And why is that important? Because the Gompertz, which we're about to learn, has a property that you can make the graph, the curve for the absolute value of dy dt non-symmetric, which is important because a number of biological processes, the way they work forces it to have a non-symmetric bell curve. My idea is that maybe if the longer a tree's been infected, the more inoculum it can be producing and whatever, and that actually forces it to become non-symmetric. So, also one last thing, we can linearize it. We can linearize. Then it natural log of y over 1 minus y each to the natural log of y naught. Okay, sometimes the linearized form is easier to work with, easier to solve for the parameter estimates. Then once you get the parameter estimates for this form, you can transform them back to the original form, whichever form you're interested in, and they can be easier to deal with. So that's that. Next one's called the Gompertz. And graphically, the Gompertz can take on a lot of different shapes, but sort of what it looks like. Something like that. It's kind of like a logistic shape, but it's definitely a different shape. And then one of the nice properties about it is you can adjust, or not y, the absolute value of dy dt, you can make it a non-symmetrical shape. So maybe for biological reasons that shape's more important, that shape's more meaningful. Then you can do that instead of having this versus a nice symmetrical thing. Then we write dy dt is equal to rate of Gompertz times y times, this is where it gets kind of weird, natural log of 1 minus natural log of y. So it's very similar setup to the logistic. In the logistic, it's the rate parameter times y times 1 minus y. So in this we have the rate parameter times y times a function of 1 minus y. Instead of doing 1 minus y, it's natural log of 1 minus natural log of y. But it's basically just a function of that. And so the rate's still a function of the diseased plants and a function of the non-diseased plants. The only difference is a function for the non-diseased plants. Term is different. And that's called the Gompertz. And if you do a bunch of algebra, you can eventually find out that y is equal to exponential of negative b e to the minus r sub gt, where b is equal to minus natural log of y sub 0. Voila. Then the last one we're going to mention is called Richard's model. And dy dt is equal to r sub r 1 minus y. That's not right. I screwed that up. Is equal to r sub r times y times 1 minus y eta minus 1 over eta minus 1. And this can do non-symmetric dy dt curves. Accounts for non-symmetric dy dt curves. Just like the Gompertz, except this can do a big range of stuff, whereas the Gompertz is only one version of asymmetry. This is multiple versions of asymmetry. This lets you adjust how much skew you want, kind of thing. The eta term does. So that's what the Richards model is. And logistics, the function, is one of the subsets of Richards. So model comparison. You have f of y and f of 1 minus y, and you have exponential and mono, log, Gompertz, Richards. So an exponential for the what it does with the diseased plants is it uses y, and the non-diseased plants it doesn't do anything. Monomolecular doesn't do anything with the diseased plants, but does do something with the healthy plants. Log does something with both. The logistic, you have that. Gompertz does something with both, but for the non-diseased plants it does this function, natural log of 1 minus natural log of y. And for Richards, it does something with both, but for the non-diseased plants, you have this function right here that lets you change the skewness of the dy dt curve. And the logistics just a subset. When eta equals 2, Richards model when eta equals 2 is equal to the logistic model. So next, gonna switch to control of polycyclic diseases, and I'm just gonna read some stuff out of the book. There's a bunch of literature in here about it. I'm not gonna go through all of it because it would eat up time galore, and I don't totally understand it. But so, summary of disease control strategy. In a monocyclic disease, which tend to have the monomolecular curve where they sort of have that shape, they all get infected at once. They are controlled by reducing the amount of inoculum or the efficacy of inoculum. In polycyclic diseases, which tend to have that exponential increase at the beginning, effective or efficient control of polycyclic diseases depends on the initial disease intensity, a function of inoculum and its efficacy. I can't say that word at all today. And the rate parameter. At low rates, reducing the y0 can be variable. At high rates, reducing y0 is not effective unless something's very large. Unless some value is very large. I gotta go back and find out what that is. But anyways, and the rate must be kept relatively low value to obtain effective control unless the time period of the epidemic is very short. Although control in polycyclic diseases emphasizes reduction in rate, it may be necessary to reduce Y naught as well. For examples, in cases where there's high values of Y naught that require reduction even in the absence of further disease progress. For example, initial disease intensity of apple scab from overwintering inoculum called primary scab can be very high, with 50% of the leaves infected, even if there was no further increase in disease intensity. The initial disease intensity would be sufficient to cause economic loss. Reducing R naught, the R value, the rate value, a small amount can have a big effect on polycyclic epidemics. This is fundamental to van der Plank's thesis on disease control. It goes into a bunch of stuff related to that. Anyways, so depending on what type of curve the disease follows, there's different suggestions on where you should focus your efforts on reducing it. You can either focus your efforts on reducing the Y naught, the initial amount of disease, or on reducing the rate, or a combination of both, or some other stuff. So that's it. Then model fitting, you collect a bunch of data, and then you do a best fit of a given model to the data. So you fit the exponential to the model of data, see if it's suitable or not. If it's not, then you fit a monomolecular model to data, see if it's suitable or not. If that's not good, you'd fit another one. And then there's ways to measure how good the fit is to the data, and then you pick the model with the best fit to the data. I'm not going to go into that. That's like a whole course in itself. And then you can compare models and compare disease progress curves and do hypothesis tests on them. And I'm not going to go into that. And that completes today's lecture. Also, before I end this today, so it's like second to last day in December, and I don't know what the temperature outside, but it's like 85 degrees and 90% humidity, and the AC is running the whole time, so the audio might not be that good, but my mind's totally blown at how hot it is down here.